Hello guys, good afternoon. We're going to start today chapter 7, which is the nervous system. And there is a lot of slide in this chapter. So some of the lower part, like the last part of this chapter, we may cover in next class with the central nervous system. So let's start with the starting topic of the nervous system. And today we will talk about basically the nervous system and the nervous tissue and the characteristic of the nervous tissue. So lecture outline for today is we'll go over an overview of the nervous system. And in the nervous system, we will talk about the neurons and the supporting cells. As you know, nervous system has the neurons which generate and transmit electrical impulse. And nervous tissue has also supporting cells. And that cells support the neuron, okay? So in this subsection, we're going to talk about the neurons and the classification of neuron based on their structure and function. And then we will talk about different kind of supporting cells, their function in the brain and spinal cord. Usually when a nerve cell damage, it does not regenerate. But when the nerve fibers, which is axons and dendrites, particularly axons, damaged, then it regenerates. So we're going to see how axons regenerates. The main function of the neurons is to generate and conduct electrical activity. And that is passed through the axons. So we're going to talk about electrical activity in axons. In the axons are extension of plasma membrane of the nerve cell. And on the plasma membrane of the nerve cell, there are gated channels, which is called ion gated channels. And through those ion gating in the axons allow sodium, potassium, calcium to get in and out. And when these ions get in and out, that changes the polarity of the neuron, and that is what electrical activity. So we're going to talk about that in detail, and that is the physiology for today in nervous system. So we're going to talk about mechanism of axon potential generation, how it is generated and how it is transmitted. Conduction of nerve impulse. So nerve impulse can pass through the nerve cell to the axons. And then axon takes the impulse away from the cell body to the other target organ, or it takes to the second neuron. So how this conduction of impulse happens in axon, that is axonal conduction. We will talk about this.
Mr. Shaw,
Yes.
one of my friends in the class is having trouble joining into the class. Like the Zoom link puts him in a separate.
No, they are trying into the, what is it called, office hour link. Can you please tell them, hey, this is class link. There is two links. One is lecture link and another is office hour link.
Okay.
Yeah. Not to join the office hour. They need to join the class link. There is lecture link, conference Zoom link, and there is office hour link. I have written that there. So let me, can you send that message, please?
Yeah, I sent it.
Okay. So two neurons are attached to each other. They have gap between. So a neuron and another neuron, there is a gap, or a neuron and another cell, there is a gap. And that gap is called synapses. So transmission of electrical impulse in a neuron and through the gap is different. Through the gap, it is through the chemical compound, which is called neurotransmitter. And we're going to talk about what are the types of synapse and what are the ways of transmission, okay? So here you go. Nervous system is divided into central nervous system and peripheral nervous system. Central nervous systems are in the center of your body. That is, in the cranial cavity, you have brain, and in the vertebral cavity, you have spinal cord. And then from the brain and spinal cord, there is extension of the nerve fibers. And then there is some neuron cell bodies outside the brain and spinal cord. That is called peripheral nervous system. So peripheral nervous system, which is PNS, contains cranial nerves and spinal nerves. There are 12 cranial nerves which exit from your cranium, from your head. And there is 31 pairs of spinal nerve which exit from your spinal cord. Apart from that, in the center peripheral nervous system, there is also, you can add plus ganglia and receptors. These are also the part of peripheral nervous system. So ganglia are if some neurons are outside the brain and spinal cord, then we call them ganglia, like the sensory ganglia, motor ganglia, autonomic ganglia. And there are some receptors which receive the stimulus, which is the part of nervous system which are outside. Okay. Apart from the in the nervous system, there are two kinds of cells: the neurons and the supporting cells. The supporting cells in the central nervous system is also called glial cells. These supporting cells in the peripheral nervous system are not called glial cells. They are called glial cells because they glue neurons together. That's why we call them glial cells. Okay. Neurons are functional unit of the nervous system, so each neuron function independently. And glial cells maintain structure, chemical homeostasis of the neurons. And glial cells or supporting cells are smaller, pretty much in size, sometimes not. And they are about 5 to 7 times more common than neurons in the brain and the spinal cord. Okay. So let's talk about neurons. As I said earlier, neurons gather and transmit information. So let's see, when your room get cold, what happens? Change in temperature is perceived by receptor in your skin. That is, information is now gathered. And then neuron transmit that information. Okay. So by responding to the stimuli, it can be hot, cold, pinching, touch, pressure, whatever a stimulus is, the smell, taste, any stimulus. And that stimulus activate your receptor, and that receptor then produce and send electrical impulse through the neuron to the brain and spinal cord. And if there is one neuron from your toe to the brain, then there is no gap in between. But if there is gap in between the neuron, then there is a neurotransmitter released in between them to connect two neurons. And that's how they release chemical message through the neurotransmitter and communicate with other neurons. Okay. So neurons, a typical neuron here. A neuron have a cell body, dendrites, and axons. So a neuron have a cell body. You can see here, this is the cell. Basically, any neurons are a cell, typical cell, all the characteristic of a cell. So if you see cell has a cell body, it has cytoplasm, organelles, nucleus, and then plasma membrane. The only difference is the plasma membrane has extension. So one big extension of plasma membrane is called axons, and several smaller extension from the cell body of the plasma membrane is called dendrites. Dendrites, if you see the arrow here, dendrites carry impulse towards the cell body, whereas you can see arrow, axons carry impulse away from the cell body. There can be other kind of neuron. You can see here, this is another neuron which has cell body, but it looks like only one cell. The axon, then it divide, becomes two, and both looks like axon. In that case, we divide them whether they are axon or dendrite by the direction of impulse. Okay, so you see, dendrites are bigger here, but dendrites are taking impulse towards the cell body. That's why we call them dendrites. An axon will take away from the cell body. Okay. Uh, Neurons. Imagine, where are neurons? Where are most of the neurons in your body?
Right.
Most of the neurons are either in the brain or your spinal cord. So the cell bodies of the neurons are in the brain or spinal cord. But sometimes cell body is the nutrition center and makes macromolecules. They are like all rough endoplasmic reticulum, all organelle function here. And these cell bodies usually lie either in the brain or spinal cord. But sometimes cell bodies are outside. Okay? When they are group of cell bodies outside the brain and spinal cord, then we call it ganglia. And if group of cell bodies are in the brain or spinal cord, particularly CNS brain, then we call them nuclei. Okay? So remember the term nucleus, plural is nuclei, ganglion singular or ganglia plural. Okay. Uh, so neurons have dendrites, receive information. They receive information and convey it to the cell body. It brings to the cell body. And then you see here between the axon and cell body, there is a small hillock that is called axon hillock. And this is the location where most of the stimulus comes here and generate action potential, and it transmitted here, because in this area there will be a lot of sodium channels which open sodium get inside and produce action potential. Okay, action conducts impulse from away from the cell body, so this way. So let's see if you touch your finger first. You close your eyes and you touch finger, you feel the touch. So how that happens? You stimulate the receptor, then you are stimulating the dendrites. Okay, then dendrites brings this information. The cell bodies are in the spinal cord or somewhere outside the spinal cord in ganglia. And then that action potential passes here, travels all the way through the axon. And then in the spinal cord or in the brain, there is another neuron which is making contact with the nerve terminal here. But there is a gap. So in that gap, what they do, they release neurotransmitter. And then that neurotransmitter binds with another neuron, dendrites, and that takes create impulse and takes to the other neuron or other organ. Okay, that's how impulse transmits. Let's talk about classification of the neurons. Neurons can be classified based on their function and their structure. So based on function, neurons are sensory or afferent neuron, motor or efferent neuron, association or interneurons. Okay, so sensory or afferent neuron conduct impulse into the CNS. So let's see if it is CNS spinal cord, and if you are touching your finger, then what happens? The dendrites are taking impulse to the sensory ganglia here in the cell body, and then the axon is taking away from the cell body to the spinal cord. So the neuron which carry impulse towards the central nervous system, towards the brain and the spinal cord, is called sensory or afferent neuron. Another neuron is motor neuron. So let's say I want to move my hand. Then what happens? My brain thinks first, then motor neuron comes from my brain, motor area, to my spinal cord, then to my spinal cord. From here, the motor neuron cell body comes here. Their dendrites, then axon is bringing here. And then the nerve terminal, axon terminal, is attached to the skeletal muscle, and a skeletal muscle then contract. So motor neuron is carrying impulse from the central nervous system to the organs or to another neuron. Here is another motor neuron you see here. This is autonomic motor neuron. You see? So motor neuron passing from the spinal cord, and then in autonomic ganglia, synapse with another neuron, postganglionic neuron, and then these axons taking the same impulse to the smooth muscle, cardiac muscle, and glands. That's why we call this autonomic motor neurons, because we don't control voluntarily. This skeletal muscle is controlled voluntarily. So this is sensory and motor neuron. The association or interneurons are somewhere in the spinal cord or in your brain, where sensory neuron goes and connect between and then go to the brain, or from the brain, the middleman neuron between the motor neurons also are association or interneurons. So association and interneurons integrate sensory nervous system activity, and they are exclusively inside the brain or spinal cord in the central nervous system. Okay. Structural classification of the neurons. There are three class of structural classification of neuron based on their structure. So one is unipolar or pseudo-unipolar. Pretty much pseudo-unipolar, we call them false unipolar, because there is a cell body, there is one pole, but it divides into two. So it looks like one, but it is two. That's why we call them pseudo-unipolar. Last time I told you the longest neurons or cell in the body are pseudo-unipolar. You see here, this is the dendrites bringing impulse towards the cell body, and then axons taking here. So this end of your pseudo-unipolar neurons are in your tip of the finger or tip of the great toe. And then it takes, and outside the spinal cord, you have cell body, which is in sensory ganglia. And from there, this axon pass all the way from the spinal cord to the brain, and then it get into your sensory area of the brain. That's why it is almost 6 feet long. Okay. Cell body sits along the side of single process, and they are sensory neurons. Bipolar neurons, pretty much, you see here, it has two poles. One is dendrite, another is axons. This kind of neurons are found in the retina, in the sensory neuron of your nose. And the most common neurons are the motor neuron. You see, motor neurons are the typical neuron which you usually see under microscope. They are most common, and they have many dendrites and only one axons. See, so motor neurons are multipolar. Now, in the nervous tissue, apart from the neurons, there are supporting or glial cells. Okay, supporting or glial cells. PNS has the PNS and CNS has different kind of glial cells or supporting cells. Okay, so let's start with the peripheral nervous system, PNS. PNS has two supporting cells. We call them Schwann cell and satellite cells. Schwann cells, you see here, this is the axons. Schwann cell, you see, this is the Schwann cell nucleus here. The Schwann cell makes fat, and then fat wrap around the axons, peripheral nervous neurons axons. See? And this myelination of the axon increase the speed of axon potential. In between the Schwann cells and myelination, there is a gap region that is called node of Ranvier. Okay. So this is, so you see one axons are covered by several Schwann cells. Another kind of supporting cells in the peripheral nervous system is satellite cells. Satellite cells are the cells which wrap around the ganglia. So you know ganglia are the group of neuron cell bodies outside the brain in the spinal cord. So let's see if there is a sensory ganglia, that means there are cell body, neuron cell body in group. 1, 2, several neuron cell bodies. And then we have supporting cell, which is satellite cells. They are around this ganglia. So let's see, this is supporting red circle longitudinal cells. They just satellite around the supporting the ganglia. And this is called satellite cells. See? So these all red cells are satellite cells. Okay. So what does satellite cell? Satellite cell covers surface of sensory and autonomic ganglia. Okay. Okay. Now, satellite cells in the central nervous system. In the central nervous system, there are 4 major types of supporting, or we also call it glial cells. They are oligodendrocytes, microglia, astrocytes, and ependymal cells. Okay. So in this diagram, you can see this is the cut section of the brain. If you see your brain is not a solid organ. It has cavity in the center. Okay. It has cavity filled with CSF, cerebrospinal fluid. Okay. So this is the brain tissue. There is the ventricle lined by ependymal cells, and then cavity containing cerebrospinal fluid in the ventricles. So we have a section of the brain and looking at supporting cells. First have the orientation of the brain tissue so you understand. Okay. So let's talk about oligodendrocytes first. Oligodendrocytes are the cells here, oligodendrocyte. Oligo means less, projection. That's why we call them oligodendrocytes. It has 1, 2, 3, 4, 5 only. So this oligodendrocytes is myelinating, putting the myelin sheath around the axons in the central nervous system. One cell can manipulate several axons. You see another oligodendrocyte, another oligodendrocytes. There are one disease of the nervous system where there is, that is called autoimmune disorder. In that autoimmune disorder, your own immune cell start killing the oligodendrocytes. So if oligodendrocyte is killed, what happened? There will be no myelination of the neurons, axons in the central nervous system. And then your neuron cells start dying. And that condition is called multiple sclerosis. Have you heard about that? Here I have written it. Multiple sclerosis is autoimmune disorder due to destruction of oligodendrocytes, and then it caused several neurological deficit in progressive fashion. This disease is very common, typical in young female, around 20 to 30 years of female. And once you have, it is like very debilitating disease. It carry for a long time, and it has unpredictable neurological symptoms. Okay. Another cell is microglia. So you see, microglia is like spider-like cells. These microglia cells are phagocytic cells. They engulf or phagocytose foreign microorganism or foreign particles if they somehow appear in the central nervous system. Third one is astrocyte. You see here, astrocyte. Astrocytes are like a star. That's why we call them astrocyte, means cells. You see, they have cell body, they have nucleus, and they have several tentacles. And these tentacles or foot process, they have perivascular feet because their foot is attached to the blood vessels of the brain, which is called cerebral blood vessels. Due to the tight attachment of the perivascular feet of the astrocyte and tight junction between the epithelial cells of these blood vessels, astrocyte contribute in forming blood-brain barrier. Okay, so astrocytes help forming blood-brain barrier. Okay. The next is the ependymal cells. Ependymal cells are the cells which lines the ventricles cavity of the brain, and they filter the blood fluid from the blood and make CSF. And that's why there is microvilli. You can see they filter and clean the blood and put the filter in the brain. That is called CSF. Characteristic of the neurons. Neurons, one neuron cell body, one dies, it is dead forever. They have lost their mitotic capacity in their embryonic period. But supporting cells, these are all supporting cells. Once they die, they can regenerate, they can multiply. So the brain tumor, any brain tumor, comes from supporting cells. None of the brain tumor or brain cancer comes from the neuron. Okay, except some peripheral nerve, which is called the neuroblastoma outside. Otherwise, all come from the supporting cells. That's why when you next time you go online, Google it and check the brain tumors names, you're going to see astrocytoma, oligodendroma, ependymoma. So you know they all, astrocytoma means the tumor coming from astrocytes. Oligodendroma means the tumor coming from oligodendrocytes. Ependymoma means the tumor coming from
Mm.
ependymal cells.
Is this because the supporting cells, they're just more common than neurons, and then they multiply more than neurons?
No, to have a cancer, any cancer in your body, let's say skin cancer, what happens? What you need to do? What cancer needs to do? The cells needs to divide in uncontrolled fashion. Yes, cells needs to divide. Neuron cannot. It has no capacity to divide one
Oh,
time. That's why they don't make any cancer. But supporting cells divide, and that's why they make cancer, or not the cancer, the tumor.
got
But
it.
the brain cancer we call brain cancer, they are the tumor. They all are benign tumor. But the brain tumors are in the brain and it damages brain cells easily. That's why we treat them like a brain cancer. Okay. That is the supporting cells and some clinical background. Okay. So supporting cells, you see oligodendrocytes. One oligodendrocytes is making myelin sheath around 1, 2, 3, 4 axons. This is the myelination of the axons of the peripheral neuron. So here is the contrast and compare and contrast. One oligodendrocytes can myelinate several axons in the central nervous system, whereas one axon is myelinated by more than one myelin, the Schwann cells. Are you following me? So in regards to myelination, the this one, the oligodendrocytes are more efficient. Okay. This is the summary. I have told all what are their location and what are their function. I talked about them, so you can go and you can read this. Sorry. Already I told you when nerve damage, their cell body is damaged forever. It never regenerate. But axons can regenerate. So what is the process of axon regeneration? Axon regeneration occurs much more readily in the peripheral nervous system than the CNS. CNS, you cannot generate the axons because there are supporting cells like oligodendrocytes which produce protein, and that inhibits the regrowth of axon tubes. And astrocyte from glial scar tissue also blocks regrowth. That's why axon regeneration is only in the peripheral nervous system. So let's see, when you cut your finger, you're cutting the skin, blood vessel, and some axons or dendrites. What happens when you cut the axon? Do you paralyze forever here? No. When your wound heals, axon from both sides come together and joins. And let's see how it joins. So here we have stage A, B, C, D, and E. So this is motor neuron cell body, and there is dendrites and there is axons. So somehow this is the skeletal muscle in your finger, and when you have a like trauma, you have cut the axon here. So distal portion of nerve fiber is cut. Okay, when it is cut, what happens? Local phagocytes comes, inflammatory process starts, and distal portion of the fiber degenerates, like it all phagocytosed. Everything is cleared first. When the house is older, what do? We break it down and clear it. So it is cleared with the phagocytosis process by eating the debris by the nearby macrophage. And then slowly what happens? Proximal end of injured nerve fibers regenerating into tube of Schwann cells. So Schwann cell, you see axon is covered by Schwann cell. So Schwann cell first come here and divide. As I told you earlier, it is Schwann cell which divides, okay? And this Schwann cell then grows, continues till it reaches the target tissue, the skeletal muscle in this case, and it forms the regrowth, regenerating tube or regeneration tube. Once regeneration tube formed, then slowly axons also start growing because cell bodies mix all the phospholipid, glycolipid to make the more plasma membrane and extend here as axons. And then ultimately it joins again the muscle, and that's why you get the muscle function again. At the beginning, your muscle will be weak, but later you will be gaining the whole strength of the muscle. Okay? I hope you guys are understanding, and I am asking you guys, once we are done with the presentation, let me know if anything should be changed during the presentation, okay? So feedback will be welcomed. Okay, so now the main function. Up to now, we have talked about the structural part of the nervous system. Now, the main function of the nervous system is to generate and to transmit action potential or electrical activity. Electrical activity in the neuron electrical activity in the neuron is called action potential. Okay. Okay, so before you go to the slide, I'm going to give you background information. So let's see, a neuron is a cell, yes? So let's see if I have a cell here. I'm going to make simple cell here. We have nucleus. And if you extend this plasma membrane of the cell, that becomes axon and dendrites. Are you guys following me? Now, what is the characteristic of a cell? So this is intracellular space of a cell, ICS, intracellular space. And this is extracellular space. This is so hard to write with this. I don't know text. I can write in text too. Okay, so ICS and ECS. Extracellular space and intracellular space. So our cells or neurons are dissolved in the fluid. There is intracellular fluid and there is extracellular fluid. And the plasma membrane of a neuron cell is semi-permeable like any other neurons, any other cells. Okay. But the amount of ions, whether it is positive ions or negative ions, are not equal in and out of the cell when the cells are resting, when the neurons are resting. Are you following me? So check it. This is the characteristic of our neurons in our body. We got as natural evolution. The inside of the cell has more negative charge. So let's see, here is negative charge inside the cell, more negative. And outside is more positive ions. Are you following me? And these plasma membrane, plasma membrane contains phospholipid bilayer and some proteins, and proteins are called channels, pumps, receptor, marker, enzymes. Here in the neuron, we're going to talk about how the channels, these protein molecules in this plasma membrane act as channels. So there are several kind of channels. Some channels are leaky channels, some channels are gated channel, they have a gate. And due to these channels, there is unequal distribution of positive and negative ions. There are more negative ions inside and there are more positive ions outside. So is this equal here? No, that means the voltage inside the cell and outside the cell are unequal. One side is positive pole and another side is negative pole. So this is called polarized. That means plasma membrane of every neuron in our body is polarized. It is more negative inside and less negative outside, or more positive outside. Are you following me? This is the tough part of the nervous system, okay? This is the physiology of nervous system. I want you to understand. you will see a lot of question, and this is what is applied in the medical field later when you talk about any medicine which blocks, like local anesthetics, general anesthesia, they all work on these channels. So I want you to learn it, okay? So this is the condition of any neuron. And if we measure what is the difference between the charge, then the net charge difference between the positive and negative is inside the cell is negative 70 millivolt MB. Are you following me? So inside the cell is negative 70 millivolt. That means inside cell, intracellular voltage is negative compared to extra. Now I'm going to ask you a question, one you guys, and then we will go. So let's see, somehow I poke my finger and I stimulate this neuron Axon's plasma membrane. Okay. So when I stimulate and I open negative channel here, what will happen? The negative ion will go out or negative ion will go inside?
Go out.
Go out. If I open sodium channel, somehow we're stimulating, which one will go in and which one will go inside?
Positive.
Positive, because positive is more outside and I'm opening positive channels. Okay, so that's how action potential generates. This is the basic idea, and then we're going to go from here. You guys got? So in summary, the resting membrane potential of an axon, which is the plasma membrane of a neuron, is negative 70 millivolt intracellular. That means inside the cell is negative 70 millivolt, more negative compared to outside. Okay. Okay, with that, let's go to next slide. Oh, I have to remove this, I think. Can you guys save when I write it on your screen or not?
Yeah.
You can see it.
Oh, thank you. Can you save it?
Hmm.
I don't think so.
Okay. So now, resting membrane potential. I talked earlier, resting membrane potential. At rest, all cells have a negative internal charge and unequal distribution ions. So why there is negative internal charge? Because large negatively charged organic molecules like protein being trapped inside the cell. Through the evolution, a lot of large particles of protein got trapped inside and they are negative charge. So if negative, you trap more negative inside, that's why there is negative inside. On top of that, there is sodium potassium pump, a protein pump in the membrane, and limited permeability keeps sodium high outside the cells. So sodium potassium pump, what they do, it has relationship when this pump start pumping in the plasma membrane, they pump 3 sodium and take 2 potassium out inside. So like if you pump 3 positive out and only take 2 positive in, what will happen? You'll have balance of positive outside more and you create negative inside. Are you following me? And then on top of that, potassium is very permeable and is high inside cell because potassium is positive and inside the cell it is always negative that attract negative charge, also attract the potassium inside. So summary is your inside the cell is more negative. Okay. Now, if any cell, if the society is polarized, let's see, let me give you this, like if if a society is very neutral, everybody is happy, nobody has biases against each other, like they all think in the same pattern, then what happens? The society will be calm. There will be not uprise or very aggressive behavior towards each other. But if the society is highly polarized, like one group think one way and another group think another way, then somebody can come and incite violence easily, because one group believes in one thing, another group believe in another thing. You can excite that society easily. Are you following me? Similarly, our neurons are polarized. One side is negative, another side is positive. So if you poke it, you can excite it. That's why neurons are excitable. And excitability means excitable cells can discharge their resting membrane potential. So if they are negative 70, from that charge they can discharge. Discharge means they can go less negative or more positive. Okay, so they can discharge resting membrane potential quickly by rapid change in permeability of ions. And usually neurons and muscle cells can do that. Apart from neuron, it is muscle cells that can do this to generate and conduct impulse. Okay, so let's see here. This is the axon here. And at a rest, this axons inside and outside. One electrode, recording electrode, is inserted inside the axon and another is outside. And then we are recording. Okay, so during the resting membrane potential, what will happen? Our rest, the membrane potential will be at the resting membrane potential negative 70 millivolt on the computer. We are connecting outer electrode to the computer. You see here? So this is resting membrane potential. Let's see if I poke here in the neuron, what happens? I'm going to open sodium channel. Okay. Once I open sodium channel, what happens? Sodium get inside the neuron. When sodium get inside, then where does this membrane potential will go from 70?
It goes up.
It goes up, negative 60, negative 40, negative 0, negative. And that going upward is depolarization. Means from polarization, you are doing its opposite. Now, if somehow open here potassium channel, where is more potassium? Inside the cell, yes? So
Yes.
usually sodium is outside more and potassium is inside more. Okay, so if I open potassium channel or chloride channel, chloride is also minus. So if we open potassium channel, what happens? Potassium will come out. When potassium comes out, what happens? From 70, you will go down.
Go down. Mm-hmm.
And that is called hyperpolarization. Okay, so membrane potential change measured by placing one electrode inside cell and one outside. Depolarization occurs when membrane becomes more positive. You see depolarization. And depolarization is always stimulation of your neuron. So when you feel pain, that is depolarization. Hyperpolarization means membrane becomes more negative than the positive. So it goes even lower than the resting membrane potential. And then sometimes what happens? So you have, when you open sodium channel, it goes up to the 30, and then sodium channel close, and then potassium channel open. So from potassium channel, when open, what happens? From here, this graph, let's see, it goes here, and then it slowly comes outside up to the resting membrane potential. That is called repolarization. But when it goes beyond the resting membrane potential, that is called hyperpolarization. Are you following me? So hyperpolarization is inhibition, whereas depolarization is stimulation. Okay. Okay. Membrane ion channels. Membrane potential change occur by inflow through the membranes, as I said earlier. Some channels normally open, some closed. on the axons membrane, potassium leakage channels are always open, and closed channels have molecular gates that can be open. So some of the channels have gate on the plasma membrane of the axons, and they open only with the change in the. So first we give stimulus, they open the change the gate, the voltage, and that change in the voltage open the gate. That's why we call them voltage gated channels, and that's why they are opened by depolarization. Voltage gated VG channels are closed in resting cells. Sodium channels are also voltage gated and also closed in resting cells. So let's see here, these are the model for the channels in the plasma membrane of axons. You see, this is channel closed at resting membrane potential. Now it is open, sorry. And once you stimulate, then it open, and once sodium is in, then temporarily this is closed during the refractory period. So this ball is just closing in. So these are just the model of channels. So let's see here. I'm gonna make a little bit longer so you can see. So here, this is the membrane, and when you give a stimulus, what happens? There is negative 70 millivolt. This is the resting membrane potential. So when you give weaker stimulus, it will not reach the threshold. Threshold is negative 50. When you give another stronger stimulus, then once it reaches the threshold, then that opens the sodium channel in the wall, and sodium is getting in. And when sodium getting in, what happens? This wave becomes depolarized. Once it reaches positive 30, then this channel is closed, and then potassium channel open. So you see, start potassium is inside the cell more, so they get out, and that's why it is repolarization. So repolarization is due to potassium out. Now it reaches the threshold, and then still some of the potassium come out, and that once it goes beyond the resting membrane potential, then we call it hyperpolarization. So what happens? Let's see here. Once you give stimulus, what happens? Voltage regulated sodium channels gets open, and then some sodium diffusion, and that opens more voltage gated sodium channel, more depolarization happens, and they open continuously more and more till it reaches 30. So if one opened voltage gated sodium channel is opening more and more, then this mechanism is negative feedback mechanism or positive feedback mechanism?
Positive.
Positive feedback mechanism. So this is another example of positive feedback mechanism. Okay. Once it reaches the higher, then what happens? Voltage gated potassium gets open, potassium diffuse out of the cells, and then less depolarization. That's why you have repolarization. Have you heard about lidocaine? Lidocaine. Lidocaine is a local anesthetic. When you go to doctor give this medicine in your teeth, gum, to numb your teeth. So how this medication work? Let me go here back. This lidocaine blocks the sodium channel. Okay. So if you block the sodium channel here on the plasma membrane, what will happen? No matter how stimulus you go, you cut your finger, sodium channel is not opening, action potential is not going. So can you feel it? No. That's how local anesthesia work. Lidocaine is L-O-I-D-O-C-I-N-E. Okay, local anesthetic. So lidocaine, an example of local anesthetic, blocks sodium channel, and that's how they work. Okay.
I have a quick question. For the numbers, the negative 70 as the millivoltage and the 30, are those like standard figures?
Yes, a standard figure.
Okay.
Yes. Yeah.
Okay. Thank you.
And these all are again explained in different stages. You guys can see here. Okay. I think I will stop here on slide 20. So this is action potential are all or none. So once action potential generate, it is full-fledged or it does not generate. It is like light bulb. When you switch, it turn on or it does not turn on. That phenomena is called the action potential. How painful, how strong is action potential means how fast action potential amplitude is, that how frequency is, that determines it. Okay, so this is all for today, and I'm going to stop here recording, then we will talk about